I'm honored to be the inaugural figure to deliver this important lecture to this very, very important audience. And I'm very much looking forward to the panels tomorrow, where I will spend time talking about the experience that I've had, which has been a parallel career, as you say, over the last 20 years, as the kind of research that I've done in stem cell biology and embryonic stem cell biology has really been about advocating for a number of important issues that really speak to the social responsibility of the scientist. And I hope that many of you will have the opportunity to come tomorrow to talk about that. And it's very timely because tomorrow afternoon I'll take a train to Washington, D.C., where I will be co-chairing a new committee for the National Academies that is the Committee on Emerging Science, Technology, and Innovation, together with Atul Butte, who's a computational biologist, and Alondra Nelson, who's a sociologist of science. We're going to really be tackling a variety of emerging biotechnologies that span the gamut from in vitro gametogenesis to ectogenesis, mind-machine interface, artificial intelligence, and the like, and really try to get a handle on how do we as a community of scientists act responsibly so that these new emerging technologies can have an ethical framework that parallels the scientific advance. So I invite you all back for that. Today I want to talk about something which is very near and dear to my heart, which is not being the dean of Harvard Medical School, but is my life as a scientist over the last 20 years. Fortunately, when I negotiated to be dean, one of the critical elements for me was being able to retain my laboratory. And even though my laboratory is but a shell of its former self, it was very, very painful to have to resign from the Howard Hughes Medical Institute. But I had to downsize my lab considerably, but fundamentally I spend a day a week where I get to re-immerse myself in scientific research. I get to reconnect with my trainees, and that is such an important part of being a scientist, and I'm just absolutely delighted to be here in part because my first PhD student, Eugene Ko, is a very distinguished spine surgeon now here in part of your very, very famous and impactful shock trauma center. Eugene was one of my first students. I was a pre-med advisor at Harvard College for many years, and Eugene was one of my undergraduate students. He was an absolutely brilliant student, and it was very easy to get him into medical school and then through graduate school, and now he's gone on to a very distinguished career. So Eugene, it's so wonderful to see you here. So you're going to hear a little bit of my sense of the history of the emergence of cell therapies. And I think we're really at an inflection point where cells as medicines are really going to transition into a major new modality. Now, one could argue that blood cell therapy is probably the oldest form of cell therapy. And one could date it back to the 1600s. So this was a report, a case study in the Philosophical Transactions published in 1667. And it reported the first attempt at blood transfusion. Now, of course, Mr. Arthur Koga was the hapless patient who was infused with lamb's blood to treat what was almost certainly a syphilitic mania. This was maybe the first, but certainly not the last example of an ill-begotten cell therapy. As you can imagine, Mr. Koga did not do particularly well. But it really talked about the sort of intrepid nature of both the investigator and the individuals who undergo these kinds of experimental therapies. I'm not sure there was a lot of informed consent at the time, but this is the beginning. And I would say that blood cell therapies have always been at the vanguard of cellular therapy, and I think they are again today. And so where I'll take you is on a bit of a history of the emergence of hematopoietic stem cell transplantation and cell therapy, and then talk about what I think are some of the cutting-edge applications of cellular therapies that derive from pluripotent stem cells. One could trace the conceptual origins of all stem cell therapies, if you will, to our deep understanding of blood stem cells. The whole notion and use of the term stem cell first appears in the literature sometime in the late 19th century. And interestingly, in the early part of the 20th century, there was already the notion that the blood system might in fact be a panoply of cells all originating from a single stem cell, the sort of seed of the hematopoietic system. Interestingly, though, one could argue that the origins of cell therapy, at least for recovery of hematopoietic function, traced to the unfortunate circumstances of World War II. Now, in World War I, there was widespread use of nitrogen mustards in biowarfare, and many of the individuals who succumbed immediately to devastating pulmonary disease, burn, skin involvement. But those who survived ended up with a dramatic degree of hematopoietic suppression, bone marrow suppression. And in the aftermath of the atomic bombs in Hiroshima and Nagasaki, it was further appreciated that many of the individuals who succumbed were succumbing from bone marrow failure. And through a series of experimental investigations that followed the interest in radiation disease and radiation poisoning, emerged the notion that you could rescue radiation-induced lethality in rodent models by the transplantation of bone marrow, suggesting that there was an active cell within the bone marrow that was responsible for hematopoietic reconstitution. That work was then translated into human clinical care by Don Thomas, who in the late 50s realized that the combination of irradiation and high-dose chemotherapy could eradicate the leukemias and lymphomas of the hematopoietic system, but to rescue those individuals, one had to transplant bone marrow. Now, these initial patients suffered a very, very horrible demise. None survived. And indeed, it was Don Thomas himself who recognized that the ignorance that underlay that clinical experiment led him to return to the laboratory, to refocus on the fundamental mechanisms of cell transplantation. Indeed, he went back and started studying bone marrow transplant in dogs and put his own experiments in human transplantation on hold. And it was only later, when there was the appreciation of tissue matching and the need for HLA and other tissue typing, that hematopoietic transplantation reemerged. And it was a combination of those insights, both the intrepid clinical investigation, but then the appreciation of the need for tissue typing, that led to the award of the Nobel Prize for Don Thomas. The scientific underpinnings of hematopoietic stem cell transplant really traces to the transformative work of Till and McCulloch. And even though Bud McCulloch has succumbed, I had the pleasure of meeting James Till recently. Their work in the radiation response led to the definition of the colony-forming unit of the spleen, here shown, as one performs bone marrow transplantation to rescue the radiation injury in a mouse model, one can perform a limiting dilution, a decreasing dose of cells, and find a very, very small dose that will lead to the detection of individual colonies in the spleen. One can actually dissect these individual colonies and look at them under the microscope, and you'll see that they harbor a diversity of blood lineages, white blood cells, red blood cells. And indeed, there was some limited degree of self-renewal of these colonies. You could transplant individual colonies into a second mouse and see a secondary colony-forming unit. And by doing mathematical analyses between dose and colony number, they led to the idea that there were individual cells in the bone marrow that had both self-renewal and multilineage differentiation potential. And that was the origins, if you will, of the modern concept of the stem cell. And indeed, all of the underpinnings of stem cell biology have been heavily influenced by our deeper understanding of hematopoietic stem cell biology. Now, another leap forward in hematopoietic stem cell biology was the application of the transformative technology of flow cytometry, which is largely the efforts of Lee and Len Herzenberg, and its application to prospective fractionation of the bone marrow, surface antigens that can distinguish multiple cell types within the bone marrow, and then the work of Jerry Spangroot and Irv Weissman led to the most highly refined definition of a hematopoietic stem cell in the mouse. But this work has, once again, I think, been the underpinning for the prospective purification of stem cells from various tissues, but still the best understood is the hematopoietic stem cell of the mouse. Some of you will recognize this young gentleman over here who was responsible for a similar type of strategy, identifying antibodies that would lead to the prospective fractionation of hematopoietic stem cells in the human. And indeed, the ability to fractionate CD34 positive cells from the dramatic variety of cells within the human bone marrow has been a major advance in being able to combine cellular fractionation, gene modification, and the combination of gene therapy in the context of hematopoietic transplantation. So this was an area that I studied both scientifically and clinically as a young student. And ultimately in my own career, I emerged as the director of the pediatric bone marrow transplant unit between the Dana-Farber and the Boston Children's Hospital. And bone marrow transplantation is a mode of curative therapy for a variety of leukemias and lymphomas, and increasingly is very successful for a variety of gene therapies. It's now almost second nature that we can cure immune deficiency and a growing number of bone marrow failure syndromes using this combination of vector-mediated gene transfer and autologous bone marrow transplantation. But very early in my career, having studied chronic myeloid leukemia, for which bone marrow transplantation really offered the best chance of cure, I was focused very early on the challenges that still exist, even with our best efforts at tissue matching. If we transplant a bone marrow, even from a sibling, let alone an unrelated donor from the general population, there's still a high risk of graft versus host disease. The small immunologic differences between the donor and the recipient lead to an immune attack that can often be fatal. And so very early in my career, I started to think about strategies for generating blood stem cells that could be universal for all donors. And the idea was, could we one day figure out a way to generate blood stem cells from any individual, thereby replacing what would be an allogeneic or foreign transplant with an autologous transplant? And we know that when you transplant your own tissues, you suffer far fewer complications, and you obviate any of the immunologic issues of mismatch. Now, I could tell you the whole history of how we went down a lot of dead ends, trying to combine embryonic stem cell biology with cloning in the late 90s. Ian Wilmot and Keith Campbell ended up cloning Daley the sheep, taking adult cells and transferring the nucleus back into an egg, regenerating embryonic development, and in fact the development of an entire organism. Just a year later, Jamie Thompson had isolated human embryonic stem cells, and one of the goals that I had very early in my scientific career was to imagine that we could put together those two technologies, somatic cell nuclear transplantation and embryonic stem cell biology, to create cloned tissues. That never proved feasible in the sense of nuclear transplantation was such a cumbersome strategy. And my laboratory very early on was also interested in the notion of somatic cell reprogramming. Ultimately, we were scooped terribly by the work of Shinya Yamanaka. Always a depressing factor to realize that someone else has won a Nobel Prize after you've been struggling in an area for years. But it was certainly an invigorating demonstration that you could take a skin or a blood cell and introduce 4 genes that were normally active in pluripotent stem cells, and through epigenetic remodeling, actually convert the differentiated state of the somatic cell back to its embryonic state, creating so-called induced pluripotent stem cells. In this case, this was work that we then rapidly translated into the long-held goal that I had of making patient-specific, autologous, induced pluripotent stem cell, patient-derived cells. That acts as not only a model for studying that disease, but also as a substrate for autologous cell therapies. My lab has subsequently generated many different models of genetic bone marrow disease, immune deficiencies, marrow failure, hemoglobinopathies. Recently, we've begun studying sickle cell anemia in induced pluripotent stem cells. And what one has now with the array of gene editing technologies, primarily CRISPR, one can do ready gene repair. So the challenge of gene therapy is bypassed now because you have immortal cells in a dish, you can rapidly correct the gene defects, and then the challenge becomes the directed differentiation of these embryonic cell types into the tissues of interest. And I'll talk a little bit about some of these differentiation platforms. But this is a system, it's an experimental system that allows for a number of interesting research and potentially clinical applications. I mentioned our recent interest in using this to study sickle cell anemia. More recently, we've been able to crack the nut of being able to configure high throughput chemical screens and genetic screens to be able to discover drugs that reverse the pathology of some of these cell types. And we recently showed how we could use this to discover small molecules that bypass the red cell defect in a disease called Diamond-Blackfan anemia. But much of this early work and a lot of the enthusiasm is still directed at the idea that we should be able to harness cells as medicine. And I'll talk a bit more about that. Now, in a slightly dated review, we've captured the large number of laboratories that have really devoted their work to the creation of lineages of blood for cellular transplantation. Red blood cells, T cells, granulocytes, macrophages, and platelets. And I'll talk a little bit about the work of some others and some collaborative work that we've done to advance some of these ends. First, I'll talk about platelets. Platelets turn out to be probably the most important early cell type that's likely to be a product that will enter patients in clinical trials as derivatives of pluripotent stem cells. There are millions of platelet transfusions a year. It's a common feature in the context of leukemia induction or bone marrow transplantation, that to get patients through the period of hemorrhagic risk, you use platelet transfusions. Now the problem is, this is a very, very problematic blood product. The units are stored at room temperature. That means they're highly susceptible to contamination, bacterial contamination. They have a short shelf life. And the platelet supplies are unpredictable. This is what happens in Boston frequently, probably slightly less frequently around the Chesapeake Bay, but you get a snowstorm, and all of a sudden that flow of donors coming into the blood donor center starts to fall off. And we end up having to ration our platelets. This was a common occurrence on our transplant service. So if you could manufacture platelets from an inexhaustible supply of cells, as you would if you could derive them from pluripotent stem cells, you could have a number of advantages. You would have a predicted supply. They would be pathogen-free and highly reliable. Think about being able to avoid any risk of pathogens like HIV or hepatitis. You could produce a better pharmacologic product that would have a better dose-response relationship. And you could engineer them from individuals who have universal type blood antigens. And this is the work that we've partnered with Koji Ito to try to anticipate the ability to translate pluripotent stem cell-derived platelets into the clinic. Koji's work takes advantage of a very clever observation, which is as you differentiate the induced pluripotent stem cells through this hematopoietic progenitor cell, you have to capture a progenitor that can be conditionally immortalized. And indeed, what Koji figured was that by engineering through a doxycycline conditional gene expression system, you can express 3 different transgenes, in this case c-Myc, BMI1, and BCL-XL, and you can capture a immortalized self-renewing megakaryocyte progenitor. Then by removing the doxycycline, which is driving these transgenes, the levels of transgene expression fall, and you get a relatively synchronous release and differentiation into platelets. These pluripotent stem cell or immortalized megakaryocyte cell line IMMKCL derived platelets look by transmission electron microscopy similar to human donor platelets. They can be activated by the same kinds of proteases that activate coagulation in other platelets. And in various rodent models, one can GFP label these, show that they'll participate in blood clot formation, and even in various hemorrhage models will staunch bleeding. So these platelets look like human platelets, they behave like human platelets, but to make a unit would be something on the order of $20,000. There's a tremendous amount of serum and cytokines, and you need about 20 liters to be able to have the equivalent of a single unit. So we began collaborating with Koji's group to use a variety of strategies to try to increase the yield of platelets. And one such strategy was to first recognize if you deposited individual clones of these immortalized megakaryocyte lines, as Torsten Slager and Arun Bond did, using this very nice multi-parameter live cell imaging system called the Cell Voyager 7000, what you can see is that the vast majority of these clones actually don't produce any platelets at all most of the time. But what you get are these small subclones that have these dramatic bursts of platelets. So one strategy is simply isolating the high producer cell lines. So there's an asynchrony and a heterogeneity that can be reduced by simple subcellular or single cell cloning. And then this platform also allows us to do chemical screens for inducers of either maturation or fragmentation. And so one can configure a high throughput cellular screen. And this is just to show you the results of one hit compound. You can see the vehicle control on the left, and on the right, you can see that this compound dramatically increases the degree of platelet fragmentation. And indeed, this combination of both cloning for high yield and chemical modulation for platelet fragmentation has led to roughly a 4 to 8-fold increase in the yield of platelets. We're not yet to the point where we have something which is commercially viable, but we're moving towards a bioengineered platform that we hope will actually be able to deliver an off-the-shelf, highly predictable platelet product. And we have an FDA submission. There's been some clinical hold to address some of the chemistry and the manufacturing process, but we anticipate that this will be a clinical trial. David Steensma at the Dana-Farber is the clinical PI, and we hope to have that underway sometime in the next 12 to 24 months. Now, why is making blood from pluripotent stem cells? I've talked about justifying it from the point of view of platelet production. But for my lab, over the last 20 years, it's been a very strong platform for studying embryonic blood development. Now, we can study blood development in the mouse, but there are interesting differences between mouse blood and human blood. And so it has, for us, given us a window on human blood development that might not have been accessible. It's also the case that in hematology, the culture and expansion of hematopoietic stem cells has for many years been a holy grail, and one that has never really truly been found. Now, there's recently, by Nakauchi and others, there's been the suggestion that maybe we can, by substituting polyvinyl alcohol for bovine serum albumin and other components in mouse stem cell culture, there's really quite compelling data that they're able to expand hematopoietic stem cells. But those techniques have not worked yet for human cells. So a number of years ago, started thinking that maybe instead of trying to culture the stem cell itself, let's go back to the progenitors. Let's culture a cell that can be widely immortalized and expandable and subject to genetic modification. The other advantage is back to the original proposal that using stem cells could allow us to generate autologous or self stem cells, and this might be another strategy for combined gene and hematopoietic transplant. The challenge, though, is engineering blood development. So why is it so hard? It turns out that blood development has many of the features of multi-stage differentiation that actually characterize a number of different tissues and have been a vexing problem for many, many laboratories that work in pluripotent stem cell biology. The fact is, pluripotent stem cells are embryonic progenitors, and the first cells that they make are cells of the embryo. And in the blood, we know that blood occurs in different waves. So actually the first blood cells that emerge from mammalian embryos actually are extraembryonic. They are part of the extraembryonic mesoderm of the yolk sac. There are, as you see in this case in the mouse, the early developing mouse have these blood islands that surround the mouse. And then as the circulation is established with the heartbeat, now you get a recirculation of the blood into the embryo. And it was thought, at least traditionally, that the blood stem cells emerged in the extraembryonic yolk sac and migrated into the embryo. We now know that to be false. In fact, the first blood cells in the blood islands we call this primitive wave, and they're a unique type of blood cell. They're actually a unique mesodermal lineage. They make embryonic globins, and there's a type of yolk sac macrophage which has now garnered a lot of interest because long-lived tissue macrophages and many of the microglial cells of the brain almost certainly emerge from this population of extraembryonic mesoderm. But those aren't the cells that you really want to make. Those aren't the cells that would be the cells for bone marrow transplantation. They emerge later. They're the definitive wave. These are the cells that come later in embryonic development. Turns out they come from a completely different mesodermal lineage, a lineage that emerges from a specialized hemogenic endothelium that lines the aorta. And these are the cells that actually have both myeloid and erythroid fate, but also lymphoid fate. These are the precursors of the adult hematopoietic stem cells. Now, for a number of years, my lab was really focused on the early blood lineages, and the default pathway for the embryonic cells are the primitive wave. But more recently, and this is largely the work of Igor Slukhin from Wisconsin and Gordon Keller from Toronto, they've learned how to pattern a distinct wave. They've patterned the definitive hemogenic endothelium, and these are the cells from which true blood exists. So if you will, in its cartoon form, there is this primitive wave that is the default pathway of pluripotent stem cells. And for a number of years, we just wondered whether we might be missing a hematopoietic stem cell, but it turns out this pathway goes directly from pluripotency to myeloerythroid fate. There is a second pathway. This is this intraembryonic presumptive definitive pathway that has as its intermediate a hemogenic endothelium. And it's patterning that hemogenic endothelium, which I'll talk about at the end. This conundrum of what was distinguishing these primitive from these definitive blood cells prompted a graduate student in my group, Linda Vo, to ask a very direct question. What were the chromatin or epigenetic modifiers that were restricting the multipotency of hematopoietic stem cells? And so she configured a screen using these myeloerythroid derivatives of pluripotent stem cells. She introduced a small hairpin library, a library that we had used previously to identify inhibitors or barriers to somatic cell reprogramming. And she simply asked, which of those enzymes limit the ability to make T cells and B cells? And she ended up in this screen identifying a histone methyltransferase, EZH1, that appeared to act as an inhibitor of multipotency. Now, EZH1 is the less well-studied partner of the polycomb repressive 2 complex, where EZH2 is better known. If one knocks out EZH2, you can be replaced by EZH1, and the histone methyltransferase complex methylates the lysine 27 on histone H3. This is the primary marker of gene silencing. But there's an alternative complex where EZH1 binds to another component of PRC2 and actually sits on active chromatin. This has been work that was demonstrated by Jan Zhu and Stu Orkin. So when we did this knockdown, we had to then address, was EZH1 working through a repressive or an activating mechanism? And so we did the standard approach of molecular analysis through single cell and bulk RNA sequencing. And if you knock down EZH1 and look at its effect on the hematopoietic gene expression program, what you see is the reactivation of a large number of immune loci and immune response genes. And then if you do another molecular analysis, which is essentially a way of looking at what parts of the chromatin are open or accessible to invasion by transposons, again, the knockdown of EZH1 identifies the activation of regions of the genome that are involved in lymphocyte function and T cell activation. That was in the human system. We were able to go back in genetically modified mice, and we were able to look at that early stage of embryonic development in that hemogenic endothelium. Here is this developing mouse embryo, and that's the aorta. And we know the aorta is lined by these endothelium that are the precursors of blood. So we could perform a similar kind of RNA sequencing and again see this signature in the absence of EZH1, signature of lymphoid development. And we could perform the same kind of ATAC sequencing or transposon accessibility and once again see this signature of hematopoietic stem cell emergence. So what this told us was that the EZH1 knockdown was releasing the otherwise silenced lymphoid program in the embryo. We then went further, because if that's the case, if EZH1 is the mechanism by which the and early gestational blood lineages are restricted from multipotency, then perhaps knocking out this restriction would lead to the precocious emergence of blood stem cells. And so blood stem cells have always been defined by their ability to transplant and rescue otherwise lethally irradiated mice. So as in this system, one can show that the first blood stem cells in the mouse emerge from this hemogenic endothelium of the aorta at about 11 and a half days of gestation. If you go back a day earlier, you can, if you do hundreds of mice, you can find the occasional cell which is emerging. So the sense is the emergence is happening sometime between these 2 days. But no one has ever been able to detect a blood stem cell at the earlier stage at 9-5, where there's really just the onlogga of the AGM region. So we decided to do the experiment of asking whether knocking out this negative regulator of hematopoietic multipotency would lead to the precocious emergence of stem cells. And I wouldn't be making such a point of this if the experiment didn't work. The experiment worked beautifully, and it was really quite remarkable that EZH1 deficiency in the embryo leads to a dramatic precocious emergence of blood stem cells, suggesting to us that indeed the ontogeny of the blood system, where the myeloid and erythroid cells emerge first and multipotency only emerges later, is a system that's regulated by this EZH1 histone methyltransferase. And indeed, we're now involved in trying to understand what are the pathways upstream of EZH1 and what are the co-regulators of EZH1 that actually underlie this developmental regulation. And then that was work that was published about 1.5 years ago. So what are the lineages, other than platelets, that I think are going to have early and important therapeutic potential? And it's T cells. Now, CAR T cells have become all the rage in cancer therapy because they've proven to be a curative strategy for lymphoid malignancies that were otherwise quite refractory. The CAR T cell strategy involves pheresing or isolating T cells by the process of leukapheresis from a patient with, let's say, acute lymphocytic leukemia, and then purifying the T cells and transducing them with a chimeric antigen receptor, typically directed against a B cell antigen like CD19, and then expanding these cells and then reinfusing them into the patient. This has proven remarkable. I mean, a disease that might only be rescuable in 10 to 20% of examples, all of a sudden now you're seeing 80 to 90% complete remission rates, and those remissions are durable. The problem is that this is a patient-specific and highly cumbersome and therefore expensive process. And the first therapies to emerge from Novartis or Kite and Gilead are charging hundreds of thousands of dollars. Now with some increased accessibility to these therapies, one would hope that there would be a democratization, there'd be a reduction in cost and an increase in access. And pluripotent stem cells, I'm going to argue, I think, are the strategy. Now, this was shown as a proof of principle by Michel Sadelain, one of the pioneers of CAR T cell therapy. What he did was taking that peripheral T cell population, which has its own rearranged T cell receptor, he performed Yamanaka type reprogramming, was able to generate a pluripotent stem cell, which he could then engineer with a chimeric antigen receptor, the CD19 CAR, and then using strategies that had been developed by Juan Carlos Zuniga-Fluker and others, could differentiate into T cells. Now, in a humanized mouse model of a Burkitt lymphoma, a classic CD19 lymphoid malignancy, this is a malignancy that in this system rapidly kills the mouse, and you can rescue the mice really dramatically with CAR T cells. And indeed, the pluripotent stem cell derived or the iPS derived CAR T cells worked, but they weren't as vigorous and robust. And when you looked at the gene expression profile, they had that classic signature of an innate T cell. They were less robust in their proliferative potential. They had generally a lower expression of CAR. And it essentially said you could make off-the-shelf T cells, but you would be best served by making a T cell that was more like an adult peripheral blood-derived CAR T cell. And that's where our EZH1 strategy comes in. So Ranjing in my lab has been able to take Linda Vo's demonstration that EZH1 can enhance the lymphoid potential of pluripotent stem cell-derived T cells, and he's come up with a strategy to generate very, very high yields of CD8 positive effector T cells, CD4 single positive effector T cells. These are T cells that express the alpha beta receptor. He can engineer them with a chimeric antigen receptor, in this case one that has a co-linked M-cherry, and one can then follow the expression of the receptor, and he's able to generate very, very high yields of effector CD8 positive CAR T bearing cells. We initially thought that engineering them with the CAR might actually provide a proliferative boost, but turns out that they grow no better than, but still quite vigorously in culture. And he's been able to then demonstrate that these cells have killing activity and degranulation activity against a target leukemia cell, in this case, model system where we've engineered the CD19 target into the K562 cell. Work is underway demonstrating that these cells also have killing activity in vivo. And I do think that it's this type of off-the-shelf T cell that's going to democratize CAR T cell therapy. And then finally, let me talk about the strategy that we've been attempting to achieve over the last even 20 years, which is to make a hematopoietic stem cell from a pluripotent stem cell. And this is the work of Ryo Sugimura, and it really is built upon the foundations laid by Gordon Keller and Igor Slukvin, who demonstrated that one could, in a sense, replicate gastrulation in a dish. If you think about the primitive streak formation of embryonic development, it's the first time in the embryo where you have polarization. You have a head distinct from a tail, you have a front distinct from a back, dorsal from ventral. And this can all be done by patterning morphogens. And indeed, in the Petri dish, we can pattern what looks like a posteriorized streak, and then develop what, for all the world, looks like the same hemogenic endothelium that is part of the developing aorta. Now, the problem is that despite great efforts from Slukvin and Keller and our lab, these immediate precursors, blood stem cells, don't engraft mice. So what Rio asked was, what are the missing factors? Are there factors? And in this case, we built upon the intellectual history of Yamanaka to capture transcription factors that might convert these cells to true engraftable hematopoietic stem cells. So we started with the hemogenic endothelium. We introduced a restricted library of transcription factors, and then did an in vivo screen, capturing from the peripheral blood of these mice cells that had engrafted. And indeed, what we could demonstrate in the marrow of these engrafted mice was the presence of human blood antigen expressing cells. In addition, we could find cells that were marked by CD34, the Kurt-Sivin antigen that is characteristic of hematopoietic stem and progenitor cells. They had human red cells, human white cells, human B cells, and T cells. And then we could rescue the engrafted blood, rescue the transgenes, and a signature, a recurrent signature of 7 transgenes appeared in multiple mice. And we could then isolate those 7 transgenes and recapitulate the ability to generate engraftable cells. And indeed, those cells could be transplanted from primary mice into secondary mice, and we could demonstrate in the marrow of secondary mice features of hematopoietic stem cells, red blood cells, white blood cells, B cells, and T cells. And looking deeper, because recalling the fact that it's lymphocyte potential that distinguishes those pluripotent stem cell derived primitive from definitive cells, we could demonstrate that the mice had in fact reconstituted their thymus with T cells. They were alpha beta positive. And when we looked at the T cell receptor, they were polyclonal. And then in an experiment that I still think is one of the most exciting, we could actually show that these mice had reconstituted human immunoglobulin, not just immunoglobulin IgM, but IgG, so suggesting that there was B and T cell cooperation. And then furthermore, we could actually inject them with ovalbumin and show that you could get a boostable immune response. So we had lymphoid function, myeloid and erythroid function. What remained was to prove that in fact some of those lineages had derived from a common ancestor, hence a hematopoietic stem cell. And so we worked with Alan Engelman, who's a maven of viral insertion analysis for clonality, and we could demonstrate in these mice some of the myeloid lymphoid lineages were in fact derived from common integration sites, hence hematopoietic stem cells. Now these stem cells then we could analyze, we could purify them from the mouse marrow and perform single cell RNA sequencing. And what you can see by this various strategies for reducing the multifactorial notion of these data, that here in red, these are single cell analyses of cord blood, and they are quite distinct from most of the pluripotent stem cell derived cells. There is a tantalizing overlap in a small subset of cells. But if we do a supervised analysis, instead supervising on the basis of 62 hematopoietic stem cell specific genes, we now start to see that the core transcriptional signature of the stem cells has quite a bit of overlap. So we're not yet able to claim that we have generated the same bone marrow derived hematopoietic stem cell, but we are tantalizingly close to a cell that has self-renewal and multilineage and hematopoietic reconstitution potential that functions the equivalent of a hematopoietic stem cell. And so where are we now? We are moving this platform into our core laboratory. We've been able to demonstrate that we can effect this reprogramming using episomally derived and delivered factors. So it doesn't require stable genetic modification. But whereas the work to date shows that we can generate enough hematopoietic stem cells to rescue a mouse, that is orders of magnitude less than we would need to be able to rescue even a small pediatric patient. So one of the strategies is going to involve scale up of this production. So let me finish and just summarize that we've focused now for many years on this platform of being able to take patients with genetic blood diseases, engineer them into a pluripotent stem cell that's given us a model for studying these diseases, and we have worked diligently to try to understand the principles that will allow us to differentiate them into various hematopoietic lineages. Now, of course, once again, this is, if it were to be applied to every individual patient, would be a very, very costly and cumbersome strategy. So I think the future involves a different approach, again, using either banks of pluripotent stem cells that come from individuals who happen to be homozygous at their HLA loci. They've inherited the same HLA from their parents, and so they represent half of the genetic complexity. Or a strategy which has been called cloaking technology, and we're working with Chad Cowan, who has engineered multiple different gene knockouts into pluripotent stem cells to eliminate the expression of class 1 or class 2 and to express mimics, class 1 mimics like HLA-E or HLA-G, in order to bypass NK cell mediated destruction. And we actually think it's going to be some combination of this cloaking technology together with the directed differentiation that's going to lead to an ability to actually engraft patients who might not otherwise have an effective bone marrow donor. So I think along the way I've tried to identify and mention a number of folks who helped in this. It's been an effort across my own lab and a number of different outstanding collaborators who are listed here. And I appreciate your attention, and I hope there will be a few moments for questions. Thank you very much.